Okay, so we're going to move on now to the next inaugural lecture. So I'm delighted to introduce Vincenzo. So Vincenzo got his medical MD degree in 1985 at Catanzaro University in Italy. He then trained in clinical pharmacology, getting his CCT in 1988. And this was followed essentially by more than 30 years experience in a whole range of translational research. Between 1990 and 98, he held MRC and Wellcome Trust grants at the School of Pharmacy, where he undertook patch clamp investigations studying cortical epileptogenesis and excitatory neuromuscular excitatory neurotransmission in cortical neurons. Between 98 and 2008, he held really quite senior positions in pharmaceutical companies, particularly Eli Lilly and GSK, allowing him to develop and build expertise in drug development and early phase clinical trials. And during this time, he was also a lecturer in clinical pharmacology at the Faculty of Brain Sciences at the University of Rome in Italy. Between 2008 and 13, he was the head of clinical studies at the NIHR Imperial Clinical Research Facility, overseeing and running the center and managing a whole portfolio of clinical studies, and was principal investigator in a number of phase 1 and 2A clinical trials, including a landmark phase 2A trial of nicotinamide-induced upregulation of frataxin in Friedreich's ataxia, published in the Lancet, and which has led to a major NIHR EME program grant. He came to Queen Square in October 2013. I'm delighted that we appointed him as the founding head of the Leonard Wolfson Experimental Medicine Center, which I think is now recognized as one of the world's leading centers of excellence in trials in neurodegeneration. He was also the founding director of the MRes in Translational Neuroscience, which has been really successful. Since April 2016, he's been the director of the entire NIHR UCLH Clinical Research Facility, and has led on the major renewal grant application, which was successful for 6.5 million. In addition, in March 2020, he became centrally involved in our COVID-19 response and oversaw the COVID-19 vaccine trial program at UCLH during the pandemic, which included 6 large-scale multiple vaccine trials, including the landmark Oxford AstraZeneca trial. Vincenzo has published widely and has many other roles in various committees, including chairing the Research Ethics Committee for London and Chelsea Committee since 2016. So it's a real pleasure to now ask Vincenzo to deliver his inaugural lecture. Hopefully you're there, Vincenzo. Great.
Thank you so much, Mike. Thanks a lot. I'll try to share my screen. So thank you so much, as I said, Mike. It's a great pleasure to give this presentation. And when I thought to the title that I would give, then I thought quite some time. I wanted to cover my journey through translational neurology, but then given my various works in different settings, then I thought whether what I contributed to was innovation or lost in translation. Hopefully, I mean, is the former, but that's where my title is coming from. So perhaps what I would do, I would start from my time in the secondary school, when I was studying about the Roman Empire and the Magna Grecia, that part of south of Italy, which underwent a significant and long and prolonged period of Greek civilization between the 8th and the 2nd century BC. And that per se, that topic would not be relevant to neither medicine nor translational neurology if it wasn't for Alcmaeon, this Calabrian pre-Socrates physician and philosopher who lived in the 5th century BC. So what Alcamion did, he introduced two revolutionary concepts for those days. And one was that the mind and soul are located in the brain rather than in the heart, as everyone thought until then. And the second concept was that physicians should draw conclusions from empirical observations. And so in a way, Alcamion rejected the idea that science depends on divine revelation. So in a way, Alcamion can be considered a father of the ancient neurosciences. And being a Calabrian myself, I was very much proud and inspired. On the day of my MD degree, I really didn't know what my career was going to look like. The only certainty I had was that I had a strong interest in clinical pharmacology and translational neurology. I had no idea how I would combine the two in the context of building a professional career. Also, in those days, a duty military service was made mandatory for all males in Italy, and so I couldn't escape it. Thankfully, I managed to do it as a medical officer rather than a soldier, but that meant doing an additional 3 months to be trained in military matters and marching. Now you can imagine a bunch of young physicians who are trained to march, not even 3 years would have been sufficient. But overall, it was a good experience and a fun experience. And on a serious note, I learned two important messages that have accompanied me throughout my career. And one is on the importance to work as a team rather than individuals. And the second one is the importance to obey to rigor, rules, regulations, the importance of loyalty and moral integrity, which all these concepts, I think, are super critical and super relevant to clinical trials. At that point, then, I was ready to start my journey. The move from my local university in the south of Italy to the Consiglio Nazionale delle Ricerche in Rome, which is the equivalent of the MRC in the UK, was a bit of a challenge. It felt going a little bit uphill, but I rapidly adapted. And then my subsequent move from there to the University of Rome, where I took up my first lectureship in clinical pharmacology, was a natural progression. And so it felt going downhill. It was when I took the decision to come to the UK, to move to the UK, that it felt definitely like climbing a big mountain. My journey started at the School of Pharmacy just around the corner in 1990. At that time, my plans were to stay in the UK for 1 year or so. But then, as often things happen in life, one knows where things start, doesn't know where they end up. And so at that time, in no way I could predict, I could imagine it, I could think that my move to London, to the UK, would have been a lifetime move. At the time, my interests were primarily in excitatory neurotransmission and the mechanisms of learning and memory processing. And so to undertake a postdoctoral fellowship in electrophysiology using intracellular and patch clamp recording techniques sounded a good match. My experiments were conducted in brain slices, rodent brain slices, typically from the piriform cortex of the hippocampus, maintained in oxygenated artificial CSF at biology pH and osmolarity. And the experiments were based on the electric stimulation of afferent fibers, typically the lateral olfactory tracts in the piriform cortex or the Schaffer collateral fibers in the hippocampus, in order to record with the sharp microelectrodes inserted into the cells, or if the experiments were on patch clamp attached to the membrane surface, to record action potentials and voltage currents through the membrane of the cell. As I said, my initial plans were to stay in London for a year or so, but then things were quite successful. So based on personal grants I had with the Wellcome Trust and the MRC, my time there was much longer. And this is an example of the type of experiments and results that I obtained from brain slices, where in the frontal cortex, as well as in many parts of the brain, neurons are organized in well-defined layers with typical pyramidal neurons with a pyramidal shape, a typical apical dendrite, and a very well-structured dendritic tree are localized in the deep layers of the cortex, whereas non-pyramidal neurons lacking these features, the apical dendrite or with the dendritic tree with the more disorganized branches are located in more superficial layers. Now, when these type of neurons, both of them are electrically stimulated, they depolarize up to firing threshold, generating action potentials. But the pyramidal neurons are far more excitable than the non-pyramidal neurons. For example, when the slides are incubated with potent depolarizing agents, such as oxytremorin M, which is a potent and selective muscarinic acetylcholic receptor agonist, cells depolarize independently of their morphology, except that non-pyramidal neurons or interneurons generate isolated spikes, whereas pyramidal neurons undergo a sustained repetitive firing, which is due to a unique potassium activated calcium conductance, which essentially generates a self-maintained epiphenomenon where each action potentials generates a post-stimulus after depolarization, which increases again calcium entrance into the cells, depolarize again the cells, and reactivate more spikes. So effectively, this mechanism continues almost indefinitely until the depolarizing agent is removed from the bath, from the solution, or the cell is artificially repolarized by injecting hyperpolarizing current into the cells. So we hypothesized that this repetitive firing might represent a reinforcing mechanism during the learning and memory processing, whereas the non-pyramidal neurons, the interneurons, might be more inhibitory neurons acting as a brake on pyramidal cells to prevent an excess, a spread of neuronal excitability, which might trigger epileptiform-like activities. So therefore, the next step was to test whether anticonvulsants could have an effect on this type of current and this repetitive firing. And indeed, when felbamate, an anticonvulsant which is known to act through several mechanisms, including inhibition of voltage-sensitive calcium and sodium channels, as well as to inhibit the excitatory neurotransmission, glutamatergic neurotransmission, and potentiate GABAergic neurotransmission. Indeed, when these sort of agents were added to a perfusion with potent glutamatergic, metabotropic glutamatergic agonist, or in magnesium-free solutions, both of which are known to produce a significant increase in neuronal excitability up to firing potential, these anticonvulsants were able to stop the repetitive firing dramatically, similarly to specific NMDA and non-NMDA AMPA kinase receptor antagonists. So the conclusion of these experiments was that the tool was a very good tool for testing preclinically a number of compounds in development for neuronal excitability, either as pro-excitation or pro-inhibition. And the tool, since our experience, was adopted by several pharmas to test, as I said, preclinically, to screen the pharmacology of various molecules prior to moving to larger clinical investments. Having spent 10 years in basic science, I felt that my medical background and aspiration to progress my career towards translational neurology could have been underused if I was not going to redirect my professional interest towards clinical trials. And that's what I did. I do not believe in destiny. I tend to believe more in coincidence. But if I look back at my past, there are a number of dots that I can connect between my past life in the industry and my present life at Queen Square. I've chosen this example to highlight the dots I'm referring to, where in those days I had clearly no idea that I was going to work in due course at Queen Square, but at that time I initiated a collaboration with the headache research group at the Institute, in those days led by Peter Gosby. And the purpose of this study was to investigate the effects of a selective adenosine A1 receptor agonist on trigeminal nociception, based on the rationale that this class of compounds act as analgesics, including in neuropathic pain and postoperative pain, through presynaptic inhibition of presynaptic release of CGRP, substance P, glutamate. And so the question was whether this class of compounds, as well as acting as generic analgesics, could have an effect also on trigeminal nociception, which was one of the top priority therapeutic areas of drug development for GSK in those days. So we use the recording of the blink reflex, which is most frequently used in conditions such as the blepharospasm, which is a condition characterized by stereotyped bilateral spasms of the orbicularis oculi muscles, which lead to progressive, to an excessive spontaneous blink, and then progressively to sustained narrowing of the eyelid. And this is well known to be triggered, to be related to an activation of the ophthalmic branch of the trigeminal nerve, which can be measured precisely with the blink reflex test, which is based on the positioning stimulating electrodes on the supraorbital area, and then the recording bilaterally from the infraorbital area at the root of the nose. Now, typically in healthy volunteers, in healthy control, the response is formed by an initial action potential, initial current, which is recorded only ipsilaterally, same side of the stimulation. And then the second one, after this, there is a much longer, although smaller in amplitude, second response, which is recorded from both sides of the face relative to the stimulation. And the first recording is directly related to the stimulation of facial nerves, whereas the second one is more a reflection of the stimulation, activation of the ophthalmic branch of the trigeminal nerve. So in healthy control, the second wave is the one which corresponds to the actual blinking, and is the one that is most important in studies investigating trigeminal nociception. So in blepharospasm, as you can see here, there is a significant increase of the second wave, and this is an important diagnostic test for, as I said, the condition of blepharospasm. In our study, adenosine A1 receptor agonist had a significant effect on the second wave, on the trigeminal specific blink reflex response, and that somehow added confidence that of the potential role of adenosine A1 receptor agonist in diseases such as, of course, blepharospasm, but also other conditions such as migraine. Of course, it's not a test that can be used as a true alternative to the clinical endpoints in an efficacy trial in patients, but it is a good pharmacodynamic model that can be used, and is used nowadays in many pharmas for testing the pharmacology of molecules and for dose selection, such that positive results on the model and negative results on the true clinical trial would disprove the hypothesis underpinning the drug development of that compound. My primary role in pharma was to further characterize or better characterize a series of amyloid tracers, PET tracers for amyloid. I don't have the time to go in details through these projects, but in essence, the goal of these projects was to have a better understanding whether these PET tracers are traits or state markers, whether, in other words, they are useful as a diagnostic tools to separate between, for example, AD patients and healthy controls, or they can be used reliably in clinical trials for testing for drug effects, for testing the effect of a drug. So we and other groups around the world have shown that that is the former. In other words, these tracers are very good in differentiating between healthy subjects and patients. Therefore, they can be used even in clinical trials for entry criteria in separating to identify the type of volunteers that meets the inclusion-exclusion criteria of the study. However, they have a very limited usefulness as true surrogate markers of drug effect and disease progression, as there is no linear correlation between level of amyloid load in the brain and disease severity. So this type of question was absolutely fundamental for a drug company involved in the development of disease-modifying agents for Alzheimer's disease, and to better understand what type of endpoints should be included in trials. And now, as you all know, I mean, this type of traces are now systematically used in clinical trials for the purpose of correctly identifying the patient population. In 2008, I moved to Imperial College, and this, without any doubt, represents the most important step in my career, because it represented a significant change. I took up the position as the head of clinical studies at the NIHR Imperial CRF, with a role quite similar to the one I have right now at the NIHR UCL Clinical Research Facility. I would like to take a moment here to acknowledge Professor Martin Wilkins, Vice Chair for Research at Imperial and Director of the Imperial CRF, because he was much more than a line manager. He was a mentor, he was a daily guidance, and most importantly, I am extremely grateful because he had a faith on me, and he delegated to me the oversight of the entire CRF and the entire portfolio of clinical trials, despite I had spent the previous 20 years in basic science and in clinical trials, but from the pharmaceutical industry perspective. So a big thank you to Martin, because without his support, certainly today's presentation would not have happened. And I would like to give you, as well as overseeing the entire portfolio, I was principal investigator of a number of studies, and in the interest of time, I could only choose 2 examples. And I've chosen 2 examples for 2 different reasons. One example in neuropathic pain in pulmonary hypertension patients. You will understand in a second what is the connection between pulmonary hypertension and neuropathic pain. Essentially, this is a condition which is fatal, is a devastating condition with the high blood pressure in the arteries of the lung, leading to cardiomyopathy, right ventricular failure, and death. There is no cure available. There are few vasoactive substances that might temporarily produce a transient improvement, partial benefit to patients. But of course, these are not disease-modifying agents. And the major drawback of these substances, these vasoactive substances, in particular, one called treprostinil sodium, which is given by subcutaneous infusion, is that it produces severe local regional pain and skin reaction by acting on prostacyclin receptors in the nerve terminals at the site of infusion. So the reason I've chosen this example is because this project was effectively driven by patients, because patients are experiencing such an unbearable pain after they receive this type of treatment subcutaneously, to the extent that even the touching of clothes to their skin produces such an unbearable pain that they are typically non-compliant. They stop the treatment even though it produces some benefit because of the severe pain. So we had the idea of testing capsaicin patches that had been recently approved for the management of pain in postherpetic neuralgia, in shingles. And the rationale is that they are selective agonists of the transient potential amyloid 1 receptors. They overstimulate the skin nociceptors to become desensitized, and a simple application, just a single application for 60 minutes, produces a long-lasting pain relief up to 3 months in postherpetic neuralgia, precisely because of this desensitization of the skin nociceptors. So we thought that it was worth trying. And so we applied this patch in a pilot study with few volunteers. And so, as expected, there was a significant reduction in pain, in treprostinil-induced pain, both subjectively on heat-cold threshold, pain threshold, and subjective allodynia hyperalgesia, but also on laser Doppler fluxometry to measure the microcirculation of the skin in the area where the drug was administered. And so there was a significant reduction. This type of study was replicated by others in the subsequent years. And interestingly, all patients, or most patients, only one did not respond in this particular study. All of them at the end of the study begged us to continue the treatment on the patches because their quality of life had improved significantly. The second example I wanted to highlight is a project in Friedreich ataxia. And the reason I'm highlighting this is because this study represented a very first attempt of an epigenetic therapeutic approach in a disease different than cancer. Friedreich ataxia is the most common inherited progressive degenerative ataxia in the UK. The onset is typically in the childhood, with a progressive loss of coordination, physical disability, cardiomyopathy, and then typically, or in most cases, volunteers or often die before reaching the adult age. And there are no disease-modifying agents or therapies available. And so the primary cause of the disease is based on the expansion of a GAA repeats within the intron 1 of the frataxin gene, which leads to heterochromatin formation, transcriptomic silencing, and deficiency of the frataxin protein, which is an essential mitochondrial protein, which is thought to act as an iron chaperone to protect against oxidative damage. So our study, which was published on The Lancet, was driven by preclinical observation that the histone deacetylsalicylase inhibitor nicotinamide, which is the commonly known vitamin B3, could remodel the pathological heterochromatin and upregulate expression of the frataxin protein. And so that's exactly what happened in our clinical study, where the administration of 2 to 8 grams of nicotinamide for up to 8 weeks, bearing in mind that 8 milligrams is a huge dose, considering that the typical dose as a vitamin supplement is in the range of half of a gram, 500 milligrams. So 2 to 8 milligrams was a huge dose. And because at the beginning of the treatment, a significant nausea and vomiting in patients, but then this rapidly adapted within a few days. So at the beginning of the study, we had to work a little bit to identify a dose which was well tolerated. But then approximately at day 5 or 6 of the treatment, all volunteers well tolerated at very high doses. And so the study did show a significant upregulation of the frataxin protein, supporting further the rationale that this type of approach might represent a novel treatment for Friedreich ataxia. Although the study was not powered for clinical efficacy, we also saw a trend toward an improvement of neurological symptoms. So that prompted us, when I say us, I mean together with Richard Festenstein at Imperial and Paolo Aggiunti at Queen Square, we applied to the NIHR for an EME grant, which was successful, to undertake a properly randomized large placebo-controlled clinical trial to test the true clinical efficacy. This study has been delayed due to COVID, but hopefully will resume soon. In June 2013, I finally joined Queen Square as the head of the new Leonard Wolfson Experimental Neurology Center, which had been established through a significant grant for capital award from the Wolfson Foundation. So the interview was extremely tough. Probably the members of the panel don't remember it. I do remember it. It was tough. It was scary because I was given the task to develop a first-class center of excellence for clinical research in neurodegeneration. Quite scaring. But nevertheless, I mean, I do remember that all the members of the panel were very friendly and smiling at me, and I felt reassured that I was sure that they were going to provide me all the support I needed to develop such a first-class center of excellence. And so I accepted with a little bit of fear, a little bit of anxiety, but lots of excitement. It was when a few weeks later, a couple of weeks later, I entered for the first time the facility, that I realized that it was far from being a first-class center of excellence in clinical research. And so at that moment, I started wondering whether the panel whether members of the panel were smiling at me because they were genuinely friendly, or because they dumped on me an almost an impossible task. But it was too late to go back and change my mind. In March 2014, we opened the facility to patients. And I'm very proud to say that 5 years later, I did meet the objective. We did meet the objective to transform a center which didn't exist into a real center of excellence in neurodegeneration. By then, more than almost 150 trials had been adopted in most of the neurodegenerative conditions. More than almost 7,000 visits in more than 3,000 patients have been conducted. So here you can see some numbers that give a sense that success was achieved. And so it was probably one of my biggest achievements, and I'm very proud of it. Under my leadership also, we built a strong portfolio of innovative antisense oligonucleotide trials, starting in HD under the leadership of Sept and then progressively in Alzheimer's disease with Kaplan-Meier, MSA, Parkinson's disease, and importantly, motor neuron disease, of which I am the principal investigator. These studies, in most cases, these molecules, in most cases, needs to be given by intrathecal administration. Given their size, their volume, it's impossible to give them IV, as they would not cross the brain barrier. So the actual trials is extremely risky and very complex. However, the rationale behind it is quite straightforward. So traditional drugs typically are developed to act on the final target. In this case, it would be the final toxic protein of a disease, whereas an ASO therapeutic approach is based, an ASO intervenes at a critical intermediate stage between DNA and proteins. And specifically, ASOs are synthetic segments of DNA designed to mirror and bind specific mRNA sequences, so that the hybrid, the DNA-RNA hybrid, can be degraded by the RNAs to prevent the excess the excess production of a toxin protein. And so the study I mentioned I'm the PI of in motor neuron disease has been conducted in patients, in ALS patients with C9orf72 mutation. And this is the most common genetic cause of ALS. It is considered to be possibly even more severe than the non-genetic causes, other forms of ALS, but irrespective of the severity, the primary cause of the disease is linked to repeated hexanucleotide repeats, which are located in between exon 1A and exon 1B of the C9 gene. So this gene contains 11 exons, so 11 different nucleotide sequences, and the one which is aberrant is between the exons 1A and 1B. So during the transcription of the gene, there is the production of three types of mRNAs, the V1 and V3, as they are immediately after or on the exon 1A, generate an RNA containing the hexanucleotide repeat, the aberrant repeats, whereas the third one, the V2, is located beyond 1A, and therefore the transcription initiated in the exon 1B generates an RNA which does not contain the aberrant repeats. So therefore, ASOs are specifically designed in these conditions to target the aberrant transcripts produced after for the repeats that are in exon 1A, so V1 and V3. So this means that the goal is to suppress the production of the toxin protein and at the same time preserve the normal function of the protein expression, so the expression of the normal proteins. So this study started as a phase 1, first-in-man phase 1 trials for dose selection and for primarily safety and tolerability in multiple cohorts. So it was a dose escalation study. Safety was confirmed in this study, and all patients have now progressed into an open label extension of the trial in order to assess, in order to establish whether there is a true clinical efficacy. And so the readout of these studies are expected in mid-2022. As Mike said in the kind introduction, I took over responsibility of the NIHR Clinical Research Facility in 2016. At that point, the Wolfson, which was funded by the Wolfson Foundation, had to be renewed, as well as that time matched with the renewal of the funding for the NIHR CRF. So I took over responsibility to merge the two facilities into a single facility at two distinct sites. We were successful. I was successful to obtain significant funding from the NIHR. So this happened with a little bit of challenge, but the facility nowadays is fully integrated. The two facilities are now fully integrated with a portfolio of more than 150 trials opened at any given time, with the main CRF in the main campus focusing on solid tumors and hemato-oncology primarily, and Queen Square continuing to be the facility dedicated to neurodegenerative studies. And as part of my translational neurology commitments, I continue to lead on the Translational Neuroscience Master, which was launched in 2014. And I also thought that I wanted to give back the training that I received in clinical trials for the benefit of other researchers by chairing the ethical committee, which I would thought is part of an overall plan to lead on clinical trials. I would like to approach the end of my presentation by touching on COVID, not so much for my happiness of being vaccinated and as well as my daughters being vaccinated and for not catching the disease until now, but for the significant efforts that have been undertaken in the last 18 months, where I took over responsibility for the oversight of the COVID-19 vaccine trial program at UCLH, starting with the pivotal trial, Oxford trial, which led to the approval in December 2020. And then since then, we adopted several trials. So at some point, it became impossible to run all these trials in the existing space. And so a third site of the CRF was opened in December 2020, which is now the COVID-19 Vaccine Trial Center, which is fully part of the CRF. And since then, we undertook 12 additional trials, large-scale trials. I'm the principal investigator of most of them, and I'm quite pleased to say that UCLH has been consistently the top recruiting site in London and one of the highest in the UK. And these type of studies are of various kinds, from first-in-man vaccine trials, such as the Imperial vaccine being tested at the UCLH CRF, into novel trials testing the potential efficacy of novel vaccines, trials investigating the effects of monoclonal antibodies as an alternative to the vaccine in individuals who might not respond to the vaccine, such as immunocompromised patients. And actually for this study, highlighted here as PREVENT, there's been just in the last couple of weeks a number of press releases highlighting that the monoclonal antibody in immunocompromised patients provided a significant protection. So indeed, there could be an alternative or an addition to individuals who do not respond to vaccines. And additional trials who are absolutely fundamental nowadays, including trials investigating the combination of different types of trials where a volunteer is receiving a first dose of one vaccine and a second dose of a different vaccine, and so to establish whether there is any difference in terms of efficacy. Combination trials with the flu vaccine to see whether there is an interaction, whether it's possible to do simultaneously COVID and flu vaccination. And importantly, a trial which is about to finish and should be reported in the next few weeks, whether when is the right time to intervene with a third dose in individuals who have received to vaccinations in the last 6 months, last year or so. So this COVID has been a disaster for so many things. I don't have to remind anyone, but it has been also a tremendous scientific output in terms of publications, more than 6 papers in the Lancet in the last 6 months, a number of preprints which are in progress. Overall, we vaccinated more than 2,000 trial volunteers and boosted them in the last 18 months. And actually, this achievement was also acknowledged by the chief scientific advisor and the head of the vaccine task force, correctly highlighting that this work was possible just because of this super team in UCLH, which is correct. It's a perfect definition. And also the reason I wanted to highlight this letter is because it's another example of reconnecting the dots between my past and my present, as Patrick Vallance was in the days I was in GSK, the head of R&D, and so I interacted with him in a different capacity, and so he came back on a different context, different scenario. So again, I do believe in coincidence rather than destiny, but I thought I would mention. So I would stop here by thanking a number of people. I would like to start with Marik Rikamara and Raj Kengar, who are respectively the lead nurse and the general manager of the facility. They started in this capacity as at the Wolves in isolation. Nowadays, they are in this role for the 3 sites. I need to say that without them, none of the success would have been possible. They have run the facility in the most very smoothly, very efficiently, very always happy, and I still don't understand why they don't tell me off for the amount of work that they take on board. I would certainly like to thank the entire team, the super team, Sir Patrick Vallance referred to. Again, without them, these trials would not have happened. Most importantly, I would like to thank the patients, because absolutely in no way we would have achieved anything without their generous and enthusiastic participation in trials. I would like to thank a number of colleagues, MPIs, UCL and UCLH, especially Mike, for being my line manager, but also for being so supportive during the process of my professorship. And my final slide is to pay a tribute to my late dad and my mom, who had as a biggest objective, strongest objective in life, the success of their own children. My lovely 2 daughters, who are by far much smarter than me. I hope they didn't attend this presentation, because otherwise they will keep reminding me this. And finally, my older brother, who is also a medic. So he was a guidance. He was a support throughout my life. He has been a support throughout my life, and also protective. As you can tell, you can see here who was the naughty of the 2. And so I think he deserved an extra picture for keeping me out of trouble during my childhood and beyond. So thank you very much for listening. I'll stop here, and thanks again for your attendance.
Great, thanks Vincenzo for a very nice lecture. I'd like to now invite Professor Martin Wilkins, Vice Dean of Research at Imperial. That's great, Martin. You're very welcome to give a vote of thanks. Thank you.
Thank you, and thank you, Vincenzo, for a great talk. It's a pleasure and a privilege to be able to recognise your promotion in a vote of thanks after your talk this evening. It's a pity that circumstances don't allow us to meet in person. I generally jump at the opportunity to meet with another clinical pharmacologist, because I sometimes think we're down to our last few breeding pairs. But there we are. We have to adapt to the times. And thinking about it, adaptation is a word I think that befits your career. I really enjoyed hearing about your journey from Italy to London and your progress through to your current cutting edge clinical studies. We first met some 12 years ago now. You were working at GSK at Harlow at the time. You'd already enjoyed a distinguished early career in academia in London and in Italy and London, as we've heard. And I think by 2008, you'd satisfied your curiosity with pharma, and you were looking to see what a return to academic life might offer. It was Paul Matthews, who will be known to this audience, who introduced us. He was working with GSK at the time. He was working from the Burlington Danes campus next to the Hammersmith Hospital. And I remain very grateful to him for making the introduction. We were looking for a recruit. I say we, it's the Hammersmith Clinical Research Facility was looking for a new director of clinical studies, someone who understood industry and who could use their connections perhaps to help us in our relationship with pharma. And as it turned out, you were the perfect match. You slipped seamlessly into our setup at Hammersmith. I say seamlessly, but you were indeed a perfect fit for the team. But you'll recall our conversations with the Royal College of Physicians when it came to getting them to recognize your clinical training in Italy. We were introduced, I think it was to Chapter 11, which is a mechanism for getting recognition for training, for getting a consultant training certificate without having to go through a training program and all the exams. And so first off, it seemed like a shortcut in terms of time, but it was no easy ride. And you had to conduct, I think what was the equivalent of a Lord of the Rings journey, collecting and translating certificates and forms, building quite a portfolio in the process before getting it eventually signed off. It's a measure of your resilience and commitment that you did all this and succeeded. And whilst all this was happening, you took charge of the clinical research facility research brief, and you started your own research. Indeed, you formed a very productive partnership early on with Richard Festenstein, a perfect marriage, I think, of complementary skills. As we heard, it led to a great publication, and that collaboration continues to this day. But one thing that does puzzle me is you managed to do this experimental study in humans, in patients indeed, on ATK, which was the funding available from the Ataxia Society. So only you know how you did that. I think it probably cost much more than that, but it's a testament to your resourcefulness that you carried it off. And you not only embraced research whilst with us, as all academics have to do these days, you took on your fair share of teaching with a master's course. And while preparing for that, you, I think, went on some courses yourself, and you met Les Hewson. You introduced me to him, and so began a very rewarding expansion of our offering for people interested in the fundamentals of clinical research. And I'm glad to see that you've started and have a very successful course of your own now at UCL. It was with some pride that I saw you move to lead a clinical research facility in another major clinical research establishment, and even more so to witness your success there. I'm sure you'll get to experience the same. You're a great mentor, and I'm sure you'll get to experience the same warm glow as I do now in due course. You've always done things with great panache, and so I thought I'd close with a physical expression of this that I thought was worthy of highlighting. And that's the waste paper basket you left behind in your office when you moved. I don't think I've come across another one like it, and maybe because I've led a very sheltered life. But I think having such a prominent waste paper basket in your office sets the scene for some interesting conversations with visitors. It remained in your old office until recently, but I've adopted it for protection as much as anything. And I'm looking after it. I'll gladly offer it back to you should you want it back. So Vincenzo, on behalf of your former home and colleagues at Hammersmith, and I know with the blessing of your colleagues at UCL, many congratulations on your promotion and best wishes for your future success. And I look forward to further collaborations in the coming months and years. Thank you.
Thank you very much, Martin, for that really nice vote of thanks. And thank you very much, Vincenzo, for your talk. And thank you to both of our speakers this evening for really nice inaugural lectures, and thanks to the audience for joining. I'm sorry we can't take you for a drink as usual, but hopefully that'll change before too long. So that brings things to a close this evening. Thank you everybody for joining, and I hope you all have a nice evening. All the best. Thank you. Bye. Bye.